As we begin the section on reproductive physiology, I'd like to spend a moment on a few reminders. As humans, we have a lot of normal variation in a lot of our body systems. There are variations in the cardiovascular system, immune system, and many others. And as such, of course, there are going to be variations in the reproductive system. This can be both in terms of reproductive structures, in terms of the anatomy, as well as the functions in terms of physiology. There really is a wide spectrum between what we think of as male, intersex, and female. We also need to keep in mind some terminology. Genotypic sex versus assigned sex at birth versus gender and others are all different concepts that are influenced by a variety of factors. In this course, for the sake of simplicity and for the time that we have allocated to reproductive physiology, we will learn about the more common physiology mechanisms. But always keep in mind that there is always variation. As we begin this lecture series on reproduction, we will look at an overview first and then continue on to common features of male reproductive physiology. Here are some of the questions that we'll be examining in more detail, as well as the relevant textbook readings. We're going to begin to talk about commonalities that are found between the sexes and some basic concepts of mitosis and meiosis. And then we will move on to male gamete production in terms of sperm and what is in semen. Then we will transition to hormones and how they regulate reproduction and look at those in more detail. In this figure, we have a picture here of a female ovum that is surrounded by male sperm that are trying to fertilize the egg. The egg and the sperm are what are known as gametes, the male and female gametes, or sex cells. So we're going to look at how they are formed, and then we're going to briefly look at how they are controlled in terms of the hypothalamic pituitary axis. So both in males and females, there are some common themes here. The first one is this idea that both males and females both produce gametes. Gametes have 23 chromosomes that are produced from the cells of the gonads. So during reproduction, what we have is gametes from each parent fused together to produce a new cell. In order to produce these gametes, the process involved is called meiosis. This is when DNA is replicated once, only once, but cells undergo division twice. What this means is that each gamete is haploid, or only has half as much genetic information. In contrast, mitosis with a T, this is the normal cell division that is seen in most somatic cells in our body. To graphically represent this, I want to show you here that meiosis differs in two very fundamental ways than mitosis. In meiosis, DNA undergoes replication, but this is followed by a series of two cell divisions, which cuts the number of chromosomes in each cell by half. This is shown on the panel on the right. So we see that in meiosis 1, we have halving of the genetic material, but then in meiosis 2, we further divide the genetic material. At this point as well, we'll see that the gametes have a random assortment of the genetic material, and it also is able to reassort the genes so that the products are a unique combination eventually of both parents' genes when we have actual fertilization. This occurs in a process called non-homologous recombination in meiosis. So some of these concepts are very basic, based on any of the biology courses that you may have had in the past. If not, then I encourage you to read up on it a bit more in our textbook. But this idea that prevails is that both males and females go through this process, and the process of meiosis produces gametes that are haploid cells. Now, in both males and females, when it comes to hormonal control of reproduction, this happens by the hypothalamic pituitary axis. In both males and females, we have some sort of stimuli, which we'll discuss, that stimulate the hypothalamus to produce GnRH. This is the gonadotropin releasing hormone. This will travel to the anterior pituitary, where in turn the anterior pituitary will produce LH, luteinizing hormone, or FSH, the follicle stimulating hormone. This is in both males and females. Now the LH and FSH will then travel to the target organs. Here now is where we begin to see some differences between the genders, and we'll describe those in more detail as we go through each system. Keep in mind that, as with other pathways in the hypothalamic pituitary axis, we have negative control, negative feedback control that we'll see in terms of regulation. There also will be some cases where we see some positive control, and that's something that we will look at in more detail together in specific examples. Now, how is it that the anterior pituitary secretes both LH and FSH in response to one hormone, GnRH? How does it know which one to produce? It depends on the pulses of the GnRH secreting neurons. So remember, because these are hypothalamic cells, they are modified neurons that are secreting GnRH through the hypothalamus. And so they have some neurological characteristics. What we see here is that while GnRH stimulates the synthesis and secretion of both FSH and LH, these processes are controlled by the size and frequency of GnRH pulses, as well as by feedback from eventual androgens and estrogens. When GnRH pulses are at low frequency, that typically leads to FSH release, whereas high frequency GnRH activity typically stimulates LH release. And so we see here on this figure, we have the electrical activity shown in black of the GnRH secreting neurons. And you'll see that every time there's a spike, we have a corresponding increase in the LH shown in the blue dots that is being secreted by the anterior pituitary. So this pulse frequency, as well as the amplitude, how strong the signal is, changes during different times of childhood and adolescence, as well as periods in the adult. And so these changes in the frequency and amplitude at the level of the hypothalamic neurons are able to control the hormone responses that are downstream. So at this point, since we've briefly simply talked about the overview of the commonalities between males and females, take a minute to see if you understand the main concepts. Read this question and pause the video if you need. So the answer in this case is C. LH stimulates the release of steroid hormones from the gonads. And we're going to look at that in more detail coming up as well. A is not correct. because GnRH does not act directly on the gonad. GnRH acts directly on the anterior pituitary. Similarly, that's why B is wrong as well, because LH and FSH are released from the anterior pituitary. And then D is wrong because GnRH is not released continually. GnRH undergoes different changes in its frequency and amplitude of release in the hypothalamus based on different conditions. So the answer is C. We are now going to talk about the anatomy that commonly develops and differentiates from individuals that are XY. This is a sagittal view, and so it is a sideways cross-section of an adult. On the posterior side, we see the reproductive system in relation to the rectum, for example. On the anterior side, we see the reproductive system in relation to the anterior structures. And we also see here the urinary bladder that sits on top of the reproductive structures. So specifically, we are going to examine what is happening inside the testes. This is the primary reproductive organ in XY individuals because this is the one that develops the gametes. And we can see here the various structures, the ducts, as well as the other accessory glands that help in the production of semen. And then all of the semen will eventually travel through the penis, through the urethra, into the external environment. Note that the urethra, in males only, is a structure that is shared by both the urinary system and the reproductive system. We're going to look at the testes because this is the main hormonal endocrine organ in the male. Here is where the gametes are produced. Male gametes are known as sperm. So if we look inside sperm, we have the seminiferous tubules. And here is where we're going to zoom in eventually and see how sperm are produced. Now, sperm are usually, when it comes to the whole male body, they're surrounded by the external environment. So the scrotum, the skin, the capsule that surrounds the testes, is external to the rest of the body. And that's because the circulation that allows of air around it helps to make the temperature a little bit cooler. There's also a complex capillary network here in this region that also cools down the testes. I'm not going to go into that in too much detail, but the point is that in order for sperm to be healthy and produce optimally, we want a temperature that's a few degrees lower than body temperature. So while normal body temperature is about 37 degrees, in the testes the temperature tends to be around 35 degrees. Sperm production takes a few months from the progenitor to eventual sperm to be produced, takes a couple of months, but a lot gets made every day, on average about 200 million per day. So these get made in the seminiferous tubules, and then they further mature here in the epididymis structure shown here. Now, when we zoom into more of a microscopic view of the testes, when you look here on the picture on the right, we actually have an electron scanning micrograph of a cross-section of a seminiferous tubules. So you'll see here the different cells that are involved in these structures. And then these tails that you see here, these are in fact the mature sperm that are being produced. Not fully mature, but the developing sperm that are at the later stages of development. So the sperm cells, as you could see in black in this diagram of the seminiferous tubules, are shown here. What happens is the spermatogonia, these are the precursors to sperm. You see them here shown in blue, and then eventually we'll walk through the process of how they begin to develop from the outside corners of these seminiferous tubules, and then eventually lead more and more inwards to the lumen, and this is where they reach their later development. You'll see that there are a number of capillaries that surround the structure but are not within the seminiferous tubule at all, and this is important. I'll speak about that in a bit. We have these large purple cells here that are denoted in purple. They're not really purple, of course, in real life. These are called Sertoli cells, also known as sustentacular cells. These cells sustain or nurse the sperm. They really support sperm development. There are also, around here, these Leydig cells, also known as interstitial cells. These cells are important because they secrete testosterone, as we will see. Now, in this course, we will use Sertoli and sustentacular interchangeably, as well as Leydig and interstitial cells interchangeably. Historically, people named new structures from the people that discovered them or for different reasons, but we're trying to move away as a field from naming structures based on the discoverers or people's names. So we're moving on to more generic terms that are more accurate in terms of the function of the cells. So I'm giving you both names because depending on the textbook or what you hear or learn about, you will see both terms. Now, once again, I want to show you the Sertoli cells and the Leydig cells, as well as the developing sperm in an even smaller section of the seminiferous tubules. I mentioned earlier that there are a lot of capillaries around, but nothing inside the seminiferous tubules. This is very important because when you think about the actual sperm cells, we'll learn that spermatogenesis occurs throughout life. And that means that one's immune system has already been developed by puberty and by adulthood, of course. So when sperm develop, as we have noted, because of the process of meiosis, sperm themselves have half the amount of genetic material than the rest of the cells in the body. So that makes them different. They also have proteins on their cell surface that are pretty novel as well, that are not seen on other organs. So these characteristics make developing sperm look different or foreign to our own immune cells. Now, the job of the immune system is to identify foreign cells and eliminate them. And so it's possible that the immune system would mount its own immune reaction against developing sperm. So in order to avoid this, the Sertoli cells here actually provide these barriers, these tight junctions between the early stages, the initial germ cells, and then the actual developing sperm. This is called the blood-testis barrier. This is to ensure that no blood cells, because the immune cells circulate in the bloodstream, this is to ensure that no immune cells ever see any developing sperm and destroy them. So this is why the blood-testis barrier is important. It's important to protect the developing sperm from the immune cells that are circulating in the bloodstream so that they don't get killed as foreign. So these tight junctions are important, and it's the Sertoli cells that create them. So how is it then that these developing cells, these sperm, will get any nutrients? Well, this is the job of the Sertoli cells. It is the Sertoli cells that almost filter and absorb any nutrients that the developing sperm will need, will basically be absorbed and taken up by Sertoli cells, and then it's the Sertoli cells that are going to sustain or nurse the developing sperm to give them what they need, as well as absorb any waste products that have to be excreted into the circulation. Now let's take a closer look at how sperm develop. This is happening inside the seminiferous tubule. Here's now the more of the outer area of it, and towards the outer ends, like the outside of the seminiferous tubule, we have new spermatogonia that are going to develop into sperm. Spermatogonia are formed during fetal development. So when the male baby is in the womb, we have these germ cells, the spermatogonia, that are first created. And what happens is they keep dividing throughout life. That is one key difference between males and females. The spermatogonia actually continue to divide through mitosis. So mitosis is going to create duplicate cells that are 2N, that are full diploid cells. Now, the spermatogonia, when it divides into two, is going to then take one of those cells and continue with meiosis. The other one is going to stick around and keep dividing so that you always have a pool of sex cells in the males. So these keep dividing up here in mitosis. Eventually, the one of the two cells that is created with each cycle is going to continue on with creating a sperm. Here you will notice on this figure the tight junction. So once this cell has committed to becoming a haploid cell, there's tight junctions that close behind it, almost like a set of fire doors, you can imagine. You go through it and then it closes behind you so that, again, the immune system doesn't attack it. So one of the spermatogonia that are divided will become what we call a primary spermatocyte. And then that's going to begin 2 cycles of meiosis. Meiosis 1 and meiosis 2. Eventually, at the end of meiosis 2, we have what we call spermatids. Notice that now they are haploid, and 4 spermatids are produced from one primary spermatocyte. The spermatids then will undergo one more step. This is called spermiogenesis. This is now when they're going to differentiate into the look of a sperm, which means that they're going to pinch off most of their cytoplasm. They're going to gain a tail, and that's how sperm forms in the end, and is pinched out into the lumen of the seminiferous tubules. If you look more closely at the structure, we see that the nucleus of the cell takes up most of the bulk of the cell. Most of the cytoplasm is gone. It forms this head-like structure. The tip of the head has these enzymes. These will be important for fertilizing the egg. We have a number of mitochondria that are at the base of the tail, This is important for providing energy for the swimming action that the sperm needs when it enters into the female reproductive tract. Once sperm are developed, first in the testes and then they mature in the epididymis, they have to pick up a number of other secretions as they are released from the male body. These secretions make up what we know as semen. So semen is only made up of sperm at about 1%. 99% of semen are these other secretions from the accessory glands. As the sperm mature from the testes to the epididymis, when it's time for their release from the body, they travel up the vas deferens, past this pair of seminal vesicles, past the prostate, and then past these bulbourethral glands and through the urethra and outside of the body. These glands here that are circled in red, the seminal vesicles, the prostate gland, and the bulbourethral glands, are exocrine glands. They're not endocrine. They do not produce hormones. They are exocrine. They produce secretions directly through ducts into the lumen of their structures. Now, these are very important to be able to deliver the sperm from the male body to the external environment and presumably into the female reproductive tract for reproduction. What are these secretions that make up all of the semen? Certainly we have a lot of water that creates the lubrication and a large volume of it. A lot of lubrication comes from mucus secretions. There are also a number of nutrients, fructose, citric acid, and so on. When you think about sperm, they are live cells that need to have a lot of energy in order to undergo their trip, their path from the male reproductive tract into the female. They need a lot of energy to swim. There are some enzymes as well. All of the pushing of the semen through the male reproductive tract and then eventually into the female is also helped out by these prostaglandins. Prostaglandins are these substances that show to aid in muscle contraction. So the structures shown here in the male as well as in the female have a lot of smooth muscle around them. And so that helps to contract the muscle and propel the contents along. The bulbourethral gland especially, even though it's such a small gland, it helps to produce an important substance. It helps to produce some buffers, some alkaline secretions that are important for neutralizing acid. Now you might think, where's this acid? Why do we need to neutralize acid? Well, when you think about the male reproductive tract, the urethra that runs through the penis is shared by not only the reproductive system, but by the urinary system as well. So urine can often at times be slightly acidic. And so we want the semen to be able to neutralize that to keep the pH at a very healthy environment for the sperm. Also, in the female reproductive tract, the vaginal environment is slightly acidic as well, and that helps to protect it against bacteria. And so we also want to neutralize the acidic environment of the vagina as well. So all of these different components of semen really ultimately help the sperm get out of the male reproductive tract, have enough energy, have a perfect pH, and have the smooth muscle cells help it move along into the female reproductive tract. Here I'm showing you another section looking directly at the anterior view of the male reproductive tract, so you can visualize these structures, specifically to understand as well that there are two testes, typically in the male. There is a pair of vas deferens. There's only one prostate gland. Two seminal vesicles as well upstream, one prostate gland, two tiny bulbourethral glands, and then the one urethra that runs through. Now, ultimately, evolutionarily, we want the semen to travel from. So we've covered a lot of material. Take a moment to see if you can answer this question. What is the name of the cells that nurture the developing spermatozoa? Take a minute and pause the video. Okay, and so the answer in this case is A, Sertoli cells, also known as sustentacular cells. They are the ones that provide the nutrients and the support matrix for the developing spermatozoa. Slide, because what we didn't talk about is the negative regulation. So recall the negative regulation pathways. So testosterone, when you have a lot of testosterone produced by the testes, we're going to have negative regulation of both FSH and LH, as well as GnRH. But one more hormone that we haven't talked about yet is inhibin. Inhibin is a hormone that, when produced by the testes, stimulates negative regulation to FSH only. But it's also one additional mechanism of negative regulation, but it does not affect LH. It only affects FSH. In this graph, we see testosterone secretion throughout life of an XY individual that has developed in a male, based on functional testes. And so we see here that if we're looking at fetal life, so before the baby is born, we have high testosterone plasma levels, as well as a spike right around birth at the neonatal phase, whose true function of this is not fully understood. Then testosterone levels in childhood seem to go down, and then we see a spike again around puberty to really develop the reproductive system, and it remains high throughout adulthood. In an aging individual, it begins to decrease with time. This depends on a number of different factors, but it still remains quite high throughout an individual's life. In this figure, we see a lot of information all put together in context into one big picture. We see here some of the common hormone secretions for an XY individual, a functional XY individual, in terms of the common functions of the Y chromosome. You'll see along the x-axis the different times in one's life, and then on the y-axis are different serum hormone levels. I will point you to the legend here because now we're looking at a number of different hormones, not just testosterone. First, let's focus on this light blue dot. T stands for testosterone. It also has with it on this line what we call INSL3. This is insulin-like factor 3. We haven't talked about it much. It is produced by Leydig cells and is involved in the normal testicular descent. So when testes are formed in the fetus, to be able to descend them to the external environment. And we're not going to talk much more about this. But I'd like you to follow this turquoise, this light turquoise dot in this dashed line. And similar to what we saw in the previous slide, we do have quite a large spike of testosterone in fetal life, a little bit more in neonates, and then lower in childhood, and then the spike in puberty. But how does this relate to other hormones? If we look at now the dark blue bands here, these dots in the solid line, this will stand for LH, and also the yellow with a solid line, this stands for FSH. And so we see, as expected, that the LH and the FSH really corresponds to what the testosterone showed us, because recall that LH and FSH is upstream of testosterone. And so we see that these two shown here, LH and FSH, in the solid lines also are showing these increases during fetal life, then a little bit of a blip in the neonate, very, very low in childhood. And then once again, they rise greatly during puberty. Inhibin is a hormone that is shown in this light beige dashed line that is thought to inhibit, as the name implies, FSH. Now, the role of inhibin in the fetal period is not well described. Any sort of the role before puberty has not really been well delineated. But what we do know is that in puberty, it plays a role in negative regulation of FSH, and we're going to talk about this as well in other lectures. Another hormone that we haven't talked about yet is anti-Müllerian hormone, AMH. This will become very clear when we are talking about our fourth lecture on sex differentiation. And so we will come back to this as we're talking about that to see why is it in an XY genotype that anti-Müllerian hormone is so high. And this will become clear in that lecture. Now, what I also want to show in this figure is the size of the testicular volume as the XY individual moves through life. And so certainly we do have the testes that are formed in a functional XY genotypic individual. That means that the Y chromosome is not mutated, and so we'll see that in another lecture, the last lecture of sex differentiation. And so from fetal life, we have the penis growth and the testes descending, and the testes are a certain size, and as they stay relatively constant throughout childhood and right before puberty, we see this increase in the volume. And right before puberty, the majority of the testicular volume is made up of Sertoli cells, whereas eventually during puberty, when spermatogenesis really takes off and the sperm begin to divide, this is then when the majority of the volume of the testicles is taken up by germ cells, the actual sperm. And so this figure really shows a really nice snapshot of the different relationships between the hormones that are involved at different times in an XY individual's life, and how it also corresponds to the structures. Now that we understand a lot of the common characteristics of an XY individual, we can go ahead and talk about some clinical applications. Well, one of the clinical applications is a set of conditions called hypogonadism. Now, hypo, that's a prefix we've seen in other lectures as well. Hypo means under or below, or essentially an underperforming structure. So in this case, a hypogonadism really means a decreased functional activity of the testes. And when the testes is not functioning, certainly that also correlates with a decreased production of androgens like testosterone, inhibin B, AMH, and it can also affect sperm production. Now, hypogonadism is a term that really is a blanket term for a number of different conditions that are the cause of the hypogonadism. Here on the left of the slide, we see a simplification of primary hypogonadism. Primary means that the primary defect is in the gonad. And so with this little symbol here, it's showing that damage, some sort of damage has occurred at the testes first. And this damage to the testes is leading to a decrease in testosterone. Now, this can be a number of different things. There can be a number of genetic variations that cause hypogonadism, or it can be injuries, inflammation, certain infections, and so on that can affect the testes. Now, when testosterone production is lower, if we recall our negative feedback loops, then we will see that there's no negative feedback regulation upstream, and hence the anterior pituitary and the hypothalamus are increasing their LH and FSH as well as the GnRH in order to try and compensate for the reduction in testosterone. Now, some of the therapeutic strategies for primary hypogonadism is primarily to basically give exogenous testosterone to the individual. Testosterone therapy. Now, secondary hypogonadism, shown here on the right, this can be other conditions that are affecting either the hypothalamic neurons that are responsible for this axis or the anterior pituitary. These can be genetic defects, cancers, other disorders that are affecting these areas, and hence if these areas are lowering the GnRH production or the LH and FSH production, then hence secondarily you're also going to have a decrease in testosterone production. In order to address the testosterone decrease, again therapeutically, one can be given exogenous testosterone. But if fertility is an issue, if somebody does want to be fertile, exogenous testosterone is also going to continue decreasing the LH and FSH. So this could affect how sperm are produced as well. And so in this case, then, there may be other ways to substitute the gonadotropin deficiency with FSH and LH. Now recall, we've seen this before, that testosterone is a steroid hormone. So it begins from cholesterol, eventually leads to progesterone, and then in the testes we have the proper enzymes that will turn it into testosterone and another subtype, DHT, that we're going to talk about soon as well. Why is this important? Now, what does testosterone and other related steroids do, such as DHT, for example? Some of them you may already be aware of in terms of sex-specific tissues. For example, we already talked about promoting spermatogenesis, as well as maintaining and stimulating secretion from the prostate and seminal vesicles, as well as maintaining the reproductive tract. But what about other functions? Certainly, testosterone has been shown to be important for increasing the male sex drive, as well as related androgens we'll see in the female do that in the female as well. And of course, testosterone, when produced, produces negative feedback effects upstream. In terms of secondary sex characteristics, it is important for male pattern of hair growth, including baldness. It also promotes muscle growth. This is interesting because you may have heard of athletes or bodybuilders that sometimes illegally take anabolic steroids. What this means is that they're often taking testosterone-like substances to mimic this effect of testosterone promoting muscle growth. Testosterone also increases sebaceous gland secretion. So these are glands that secrete some secretions that often have distinct odors, not just in in humans, but a number of different male mammals as well. Odor can be important in terms of a male making its mark on a specific territory, for example, and for attracting a female mate. Non-reproductive, it promotes protein synthesis as well as erythropoiesis. This makes sense. This is the making of new blood, because if you're building protein, if you're building muscle, you certainly need more blood to be able to carry the oxygen to those tissues. It's also been shown that testosterone increases male aggression. So sometimes we see that as a side effect as well in those individuals that take a lot of exogenous steroids. Now, when it comes to DHT, the related steroid that is converted from testosterone by 5-alpha reductase to dihydrotestosterone, or DHT for short, that's important for the function of the prostate and also maintaining the external male genitalia and development and maintaining it as well. Also been implicated in baldness. So let's look at an application of that. So there are times when the alpha reductase, this enzyme that converts testosterone to DHT, if that is blocked, then certainly you don't have DHT anymore. Some companies are using this as a target to treat different conditions that DHT is involved in. For example, since DHT is important for prostate growth, abnormal prostate enlargement can sometimes be treated by blocking 5-alpha reductase, because the idea is if you block DHT from being formed, then you could potentially block the accumulation of DHT in the prostate tissue, and that will lead to not as much growth of the prostate tissue. There's another application for baldness, for example. Since this DHT might be implicated in baldness, there are companies that are blocking 5-alpha reductase that are showing increased hair growth, for example, in males. This drug called Propecia, for example, from Merck, at the start of the treatment, after 2 years of blocking DHT, you can eventually potentially correct some of the baldness of that is related to DHT. It's certainly not perfect, and there are a lot of side effects such as depression, loss of libido, because remember, these are important for sex drive too. So blocking DHT can have some other effects as well. So far, this was a brief overview of male reproductive physiology in terms of spermatogenesis as well as the hormonal regulation.